It's my pleasure to introduce Jeffrey Townsend as the today's Grand Round speaker. Jeff is the Elihu Professor of Biostatistics and Professor of Ecology and Evolutionary Biology, and the co-leader of the Genetics, Genomics, and Epigenetics program at Yale Cancer Center. He received his PhD in organic chemistry and evolutionary biology at Harvard University, and in 2019 received a prestigious membership in the Connecticut Academy of Sciences and Engineering for his work in developing innovative tools to study population biology, including evolution of antimicrobial resistance, disease evolution and transmission, and evolution of tumor biology, tumor genesis. His research enabled curtailment of pathogen evolution, outbreak mitigation, and used to inform therapeutic approaches in cancer metastasis. So in recognition of his prominence in the field, in 2021, Jeff was selected as the co-chair-elect of the Cancer Evolution Working Group by the AACR. And his lab is currently working on many projects, including developing bioinformatics tools for cancer genetics, epigenetics, epidemiology, and nonlinear mathematical models of disease epidemiology. So it's my pleasure to give the podium to Jeff, and we look forward to hear your presentation.
Thank you, Laos, for that wonderful introduction, and thank you and Ken for the encouragement to present today for this audience, and thank you all for basically the opportunity to present the kind of work that we've been doing in my lab. The title of my talk is "Why Me? The Mutagenic Origins of Cancer for Individual Tumors and Tumor Types," and I'm going to spend some time talking about that title, but first let me just go by my disclosure slide. I have done consulting for Black Diamond Therapeutics and Agios Pharmaceuticals. And so this title, Why Me, just was inspired by the fact that as I started working on this work, originally largely with Vincent Cannataro, who you'll see a picture of later, we realized that what we were doing to try to understand just what individual variants were contributing to cancer actually, to some degree, and the degree to which it addresses it, I'd love for you to think about as I give this talk, answers the question for an individual patient, what the causation of their individual cancer was. And I'll go through a lot of detail about that, but that gets down to the mutagenic origins, again, not the physiological origins, but mutagenic origins of cancer for individual tumors and tumor types. And I think this is it's obviously of interest to anyone who studies cancer, what the mutagenic origins of cancer are, and certainly of interest in one way or another to patients who have come down with cancer. It has been widely reported that one of the most difficult questions that patients and doctors struggle with upon diagnosis of cancer is the question, why me? Why was I struck with this ailment? And it's natural for patients to want to understand the causes behind their calamities, and it's difficult to hear only statistics and probabilities as a response. So the traditional way that you answer this question of why me is to say, well, did you smoke? That elevates your probability. Do you have this genetic predisposition? That elevates your probability. You know, how old are you? What is your ethnic background? There's lots of different predictors for whether or not someone might come down with cancer, but those aren't answers about why you came down with your cancer. Those are answers about generalizations about your life. So to date, these statistics and probabilities are nearly the only answer that science and medicine has been able to give. And one answer that's sort of straightforward and obvious, and if you are a proponent of sort of the genetic evolutionary model of what makes cancer happen, is that mutations happen, and that's why you have your cancer. It's a very general answer, though it's not terribly satisfying, but it can be broken down into a lot of different kinds of mutations. So there are clock-like endogenous mutations and processes that fuel mutation throughout the body over a lifetime. So as your body ages, you get these mutations that happen simply because the cellular processes that reproduce your DNA are not perfectly designed to reproduce it perfectly. And they can't be just because of the third law of thermodynamics. So there are endogenous processes that fuel mutation in your body throughout your lifetime. There are also mutational processes that are fueled by exogenous sources, such as viral infection-inducing apobec activity. So viral infection can cause your cell to react in certain ways, maybe for cellular defense. And those mutations that are brought about as sort of secondary consequences of your response to viral infection can also lead to cancer. And a third category is exogenous mutagenic sources, such as tobacco smoke that may affect your lungs or your head and neck, or UV radiation that can affect your skin. So these are all sources of mutation that we know about, and probabilistically we can tell patients about the fact that, you know, exposing yourself to a lot of sun may increase your risk for melanoma. If you have a melanoma, it may be partly due to the fact that you exposed yourself to a lot of sunlight at some point during your life. Now, one of the, I think, quite revolutionary sort of discoveries of recent times was that we can actually trace a lot of those sources, I should say, certainly all the sources I just mentioned, but many others as well, to when we sequence a tumor, for instance, we can trace signatures of those different sources in the DNA mutations that happen. So certain DNA mutations are more frequent, and I'll explain this in more detail a little later. Certain mutations are a little more frequent when you have a UV mutagenesis, and other mutations are more frequent when you have just simple aging processes, et cetera. And it turns out that there are enough mutations in a typical tumor that you can do a sort of machine learning deconvolution, and I won't go into the detail about that, to sort of figure out for a given tumor what were the different sources that contributed these mutations. And this is really, really extraordinary that we can figure that out. And just to give you a little bit more of a sort of a more detail on that. So here's SPS1, which is typically, it's called deamination of 5-methylcytosine, and that's considered to be sort of an endogenous aging process that sort of occurs without any particular cause other than time passing for our body through development. SPS2 is one of two signatures that we associate with apobec activity. There's defective homologous recombination DNA repair, which may be mutation-based and therefore endogenous, but related to a very specific process that might be treatable. Tobacco smoke, which you can see, of course, largely affects lung cancer, but you can also see some for liver, head, et cetera, kidney. There's some other sources, tobacco smoking. So an SPS5, which also you see as large circles here, that's another signature that has been traced essentially to aging processes, although it's a little less well understood what the underlying basis of it is. It's very clear that age is highly correlated with the amount of SPS5 mutation you get. Defective DNA mismatch repair, ultraviolet light, et cetera. And you can see these distribute themselves differently for different types of cancer. And so again, this is very consistent with what we knew already, and very consistent with what we generally did, which was say predictably, like if you have a lot of exposure to sun, you're more likely to get UV exposure, and that UV exposure then is more likely to translate to mutations that that may or may not cause melanoma, but once you have those mutations there, you know, they may cause that. So this is great. We've got the ability to see the sort of the trace or exposure in cells to these mutagens. The one thing that's missing though is that the extent to which each of those processes actually contribute to tumorigenesis still remains unknown. So we can look at what mutations are in the genome, but if I count up mutations in the genome, here's one, here's one, here's one, here's one, that doesn't tell me how much of each of those mutations are actually contributing to tumorigenesis. In fact, most of those mutations are not contributing to tumorigenesis, and most analyses find that there's only a few mutations that are contributing at a significant level, sort of at this SNV, single nucleotide variant level, to tumorigenesis. So we really need to have more in our, another tool in our plate to figure out what the level each of these endogenous and exogenous processes are contributing to a given cancer. And here's just a schematic for this, right? So the mutation 1, mutation 2, up to mutation N, however many there are that are really affecting cancer, they can cause increased cellular proliferation and survival. And sunlight may be contributing to UV radiation, may be contributing to some of those mutations more than others, because certain mutations are caused by sunlight and other ones are not. Similarly, aging may contribute to some of those mutations more than others. And what I've got right here is, if you take nothing else from this lecture, this is the main thing that I want to emphasize, is that there's sort of two stages to this. One is, what mutagens have you been exposed to in contributing to the set of mutations that are causing your cancer? And the second is, how much do each of those mutations actually contribute to the increased cellular proliferation and survival that is the phenotype of cancer? And there's a way to figure this out. But to figure it out, we need to sort of deconvolve something. And this is an old idea, and I'm going to go through it in some detail just to make sure that it's clear to everyone, that cancers are the outcome of an evolutionary process that's driven by mutation, consequent genetic variation created by that mutation, and natural selection for the more oncogenic variants. This is from Peter Knowles' 1976 Science article, a very well-known article where he just went through the idea that, it's an evolutionary process that actually produces malignancies. And in this depiction, you can see cellular lineages differentiating and dividing. You see a lot of lineages that are hashed out here, meaning they go extinct. And that's the selective process in operation. Most of our cells are all dying at the same rate as they're dividing, typically as an adult. So there's a lot of death going on. We don't usually emphasize that, but so that death may be going on. And what happens is that at some point you get lineages that are reproducing a lot more than they are dying. And those ones, in the case that they cause difficulties for your life, are usually referred to as malignancies, especially if they can then migrate to other locations. And this, so these later evolved lineages are usually the product of a series of mutations that come along during this evolutionary process. And what's happening with those mutations is they're actually enabling the cells to survive and proliferate better. So they're selected, is the terminology we use in evolutionary biology, and they persist. And that arising of those mutations within individual cells, within cancer lineages, is what we need to sort of understand, because there's two processes going on here. One is the appearance of these mutations, and then there's the amount that they actually increase the survival and replication of the cells. So to quantify cancer effect size, which what I typically call this, the effect on, on cells of actually leading to cancer, which in evolutionary biology, we just call a selection coefficient. We need to understand what the prevalence in a patient population is of a tumor. And we need to deconvolve that prevalence into two factors, because when we see a certain mutation very commonly in a kind of cancer, that doesn't mean it's causing a lot of the cancer. It may just be that the mutation rate is very high. And we've seen that very frequently in instances where we have genes that are very large or have very high mutation rates that show up frequently when we sequence tumors, but are not significant causers of cancer. And so we really need to understand, which ones are actually contributing to cancer and which ones are just typically contributing to prevalence because of an underlying mutation rate. So to quantify the cancer effective size, we have to do a fairly straightforward thing, which is take that prevalence, how frequent we see them in patients, and deconvolve it into the baseline mutation rate, how frequently the mutations are occurring in the lineage, and into the degree of selection for that mutation in the cancer lineage. And if we can differentiate those two things, then we can better understand how much is that mutation there is because of the underlying mutations that are happening across your whole genome that aren't necessarily relevant, and how much is due to those individual mutations actually increasing the proliferation and survival of the cells. So here's just a schematic of that. This is just basic evolutionary biology 101. Mutation creates variation, symbolized by the different shades of gray there. Unfavorable mutations are selected against. Reproduction and mutation occur, and the favorable mutations are more likely to survive and reproduce. And the point of this is that both the mutation rate and the extent to which they contribute to survival and reproduction contribute to what you see as an end product of the process of cellular differentiation, especially into cancers. All right, so how do we figure out that baseline mutation rate? Well, it's already been done for me anyway. It was a lot of the work was already done, which is really great, by Lawrence and others. This is a 2013 paper, quite a while ago, where they showed that mutation rate varies widely across the genome and correlates with DNA replication time and expression level. So there's these covariates. I'm not going to go into a lot of detail about this. I've talked about this before with various audiences here, but that mutation rate varies and correlates with DNA replication time and expression level, with heterochromatin marks, a bunch of other correlates that we can actually get about individual tumors. Those allow us to ask questions about, a given gene and whether or not it's got a very high mutation rate or a low mutation rate, by using those correlates to help us predict that, along with synonymous changes in the genome, which we can presume don't have any effect on the proliferation and survival of cells. So for instance, olfactory receptors, which early on were this bugaboo that would show up when we did this tumor sequencing, happen to be in a part of the genome that gets a very, very high mutation rate. It's basically not expressed, and not expressed parts of the genome don't have transcription-enabled repair, et cetera. CSMD3 is another example where there's very high levels, high correlates, and also very high mutation rate. And typically it's not considered to be a driver, even though you see it a lot in cancer tumor sequencing. And you can do regressions on this. And then I'm just going to very quickly mention that this wonderful work was done by Lawrence, but typically that work was only applied to the question of whether or not genes were overburdened with mutations. So in other words, they got these mutation rates and they just said, well, is it more than we expect? And then they calculated a p-value for whether we should put this gene in the category of mutated or not. And then they leave behind that mutation rate and then just look at prevalence in most of the analyses that were done from 2013 through 2018 or so. So typically that was sort of left behind at that point. And that's what Vincent Cannataro and I in 2018 sort of picked up on and said, look, this mutation rate is more important than for just calculating p-values. It's actually important for calculating the effect. In a biostatistician's mind, p-value is sort of a secondary thing. First, you calculate the effect of the thing you're looking at, and then you calculate p-value to see whether you should trust that effect. And so that's what Vincent Cannataro and I did. And just here's a sort of a brief introduction to how we do that calculation by convolving the gene-based rates from the silent sites and covariates with their trinucleotide context. So you can just go through tumor sequence data and you can look at what the underlying mutation rate is using basically that Lawrence et al. Approach that I talked about with the covariates. You can sort of look at every gene in the genome and calculate what the mutation rate is. And this is just one of these plots. It's just scatter plot on one axis of what the different gene rates are. And you can see there's quite a wide range here. And I think that's the most important lesson of this little image, is that the mutation rate varies quite extensively from gene to gene, from 10 to the -2 to the 10 to the -4 in this particular instance. So that's 2 orders of magnitude rate variation in mutation rate. So when you see, one gene mutated in a cancer tumor cohort at 100 copies out of 1,000, and another at 10 out of 1,000, that's only 1 order of magnitude difference in prevalence. And you can explain that by just half of this mutation rate diagram. In other words, mutation rate can explain a lot of the differences in how prevalent genes are when you look in a patient population. So you shouldn't take that prevalence as an indicator, as a strong indicator of how important a gene is in the cancer. You really need to basically understand this underlying mutation rate as well. And so then you can take different genes that are on that, that diagram, and you can look at each individual tumor and you can map out what the trinucleotide rates are. So this rate, the rate above, is just the rate at which the gene itself gets mutated. But if we want to look at every given site, the important thing is that the different mutational processes that I mentioned earlier in this talk affect different sites at different frequencies. I have a question right there. Say, is the mutation rate itself? In this case, it is, yes. Yeah. So these different mutational processes contribute differently. So in this case, I'm looking at lung cancer, which is why we have Actinin-B, KRAS, and EGFR highlighted here. And in lung cancer, you get a lot of these C to A mutations that are preceded by a T and followed by an A, and also ones that are preceded by a G and followed by an A and C and an A and an A and an A. So all of these bright red trinucleotide contexts get much more mutation than other ones. And again, I just want to emphasize that the coloration here is scaled to how often we see it. And so you see almost an order of magnitude, sometimes more with some cancer types, of variation again in how frequently given sites get mutated over other sites. So when you combine this plus the gene by gene rates, you're talking about 3 orders of magnitude, maybe even 4 in some cases, between a given site and another site in the genome and how frequently it gets mutated. So this is a really important factor to take into consideration when wondering whether or not a given site is important for driving cancer. And what you can do is you can basically tape this map and look at each gene. And here I'm looking at like an excerpt of a tiny little part of the genome. Sorry, this is site 850 to 870 in EGFR. Here's site 1 to 20 in KRAS, and here's site 30 to 50 in catenin B1. And I just want to mention that if you take these rates and then you make sure that the individual site rates are accommodated by ensuring that TCA is much more frequent than CCG chaining to an A, and do all of the math. That's very straightforward here, but a bit of a lot of accounting bioinformatically, and then map it through the actual genetic code. So you're looking at every single site in that protein and saying, well, how likely is this 850 histidine to change based on its 3 codon sites into a tyrosine or a proline or a phenylalanine, et cetera, et cetera. And some sites, of course, some changes, of course, can't really happen through a single nucleotide mutation. Others can in multiple ways, et cetera. So there's a lot of addition to add up here. But once you add it all up, this diagram tells you how likely each different change is to happen by neutral mutation. That is when we just expect new mutations to be sprayed on there and have no difference in the replication and survival. So then we get this diagram of how much each amino acid position would be expected to be mutated. And then we can compare that to what's actually observed. What's actually observed is a much more rarefied set of mutations than what you actually expect based on neutral evolution. And that's because when we sample tumors, we're sampling tumors that have been under selection for very specific mutations. And because right here, I've selected sites that actually do have an effect on proliferation and survival. So on the top, EGFR 858 leucine is a very well-known mutational site. The KRAS G12 is also a very well-known one. And then this part of catenin B1, it's a domain that is known to be oncogenic when it gets mutated. Slightly lower level in terms of the others, but the whole region across here is sort of known to be important to oncogenesis. And so what you can basically do is take the prevalence that we see, and this is in a very crude terms, and there's some corrections that are involved I'm not going to go into, but essentially divide the observed here by the expected block on the same plot on the left, and that gives you a metric for the cancer effect, or how strongly that site is mutated. How strongly that site is selected once it is mutated. And as I said, these are well-known sites in these particular cancers. And if you do that across all the different sites that you can look at, what you see is a distribution that looks like this, where on the x-axis is the cancer effect size. It ranges from 10 to the 0 to 10 to the 6, maybe even a little bit more typically. And why is that? What does this range mean? The range is what it is because that has, it's complicated, and I don't want to go into a lot of detail, but population genetically, it has to do with the population size of the cancer, the reproductive population size, how many cells in the cancer could possibly reproduce. I'm not going to go into more saying about that, but that's why it exists across this wide range. The density here is just, I'm just going to density plot across cancer effect size of these different mutations. So most of the mutations lie at this very low range where it's not even clear necessarily if they're under any selection, below say 10 to the 4 or so. And in blue, I show you the synonymous mutations, and in red, the non-synonymous mutations. So there's just a slight bias over the synonymous mutations of non-synonymous mutations to be oncogenic, but the really important mutations are all out on this tail here. And I've just shown 2 here for reference. Here's a P53 mutation that's quite common. Here's an NFE2L2 mutation that's quite common in lung squamous cell carcinoma. So these tail mutations are the ones that are important. And this harks back to what I was saying earlier. When we say, oh, lots of mutations are happening in the genome because of UV light or something like that, if they're not these key mutations out here on the tail, they're not contributing much to cancer. So we really need that component to be included if we want to ask the question, what is causing cancer in an individual tumor in an individual patient? You can do this diagram not just for lung cancer, but for lots of different cancers, and we see very much the same pattern. Okay, now just to provide you a little bit of perhaps validation that this cancer effect, is meaningful. Probably many of you are familiar with ClinVar variants that have been attributed over time with some clinical significance. And these, by the way, these are ClinVar variants that were attributed significance, not potential, not ones that weren't attributed significance. And on the x-axis, we've sort of divided those ClinVar variants up in some categories I'll talk about, but on the y-axis is the scaled selection coefficient. And generally, there's basically two comparisons I really want to emphasize here. If we look at ClinVar single nucleotide variants that are recurrent within cancer type, and compare it to other single nucleotide variants that are recurrent within cancer type, we see that the ClinVar variants have a much, much higher distribution of selection coefficient than the ones that are other SNVs. So in other words, there's, this is saying that ClinVar predicts cancer effect, but the opposite is true, and I'll show you that in the next slide. And then we can also compare ClinVar SNVs that are a single hit within a cancer type. That is ones that we only see once when they're ClinVar single nucleotide variants. Versus other SNVs that are single hit. And you can see that the cancer effect size of those ones that are, known oncogenic or believed oncogenic variants have a much higher cancer effect than the ones that are not believed to be oncogenic variants. And this is highly significant from a statistical significance point of view. By the way, this is work of Jeffrey Mandel, who's sitting over here in the audience, a grad student in my lab. And so that should be reassuring. Furthermore, if you take the mean or the top cancer effect of a given variant, they're much stronger predictions of ClinVar status than the CIF score, the polyphen-2 score, or variant prevalence, any of these measures that are typically used to try to say whether a variant is important or not. So really, you should be using cancer effect if you want to know whether a variant's important. This is also work by Jeff Mandel. Okay, so hopefully I persuaded you that cancer effect is a measure that you should be thoughtful about and use in the research you're doing. But what we wanted to get to from the beginning of this talk was the extent to which each of those processes contribute to tumor genesis. So if you'll at least walk with me on the idea that cancer effect quantifies the degree to which a given variant contributes to tumor genesis, then that gives us the key to finish that association I said. So we know, from Alexandrov's work, the degree to which, we know from this work, the degree to which mutations contribute to the increased cellular proliferation and survival. And we know from Alexandrov's work and others, Serena Nick-Zanal and others, what the contribution of various mutagenic processes toward creating those mutations are. And so by putting those two things together, we can understand the relationship between these increased cellular proliferation and survival and the actual processes underlying these mutational effects. So just going back again to Alexandrov's work, we know each signature contributes differentially to mutation counts observed in each cancer type. I showed this slide earlier. And here's the slide where you can sort of like fade out for a moment if you want, and then come back in a moment. It's only saying what I'm going to narrate through for those of you who are really interested how we actually calculate this process. But if you've understood everything before, there's nothing new here. It's just the bookkeeping of how we calculate this process. And the point is that for each source of mutation, here's deamination with age, apobec, tobacco, and then unknown clock-like signature, which were the four sources that came out of the deconvolution for a particular tumor in the TCGA data set that turned out to be useful for illustration of this. For each of those processes, there's a weight of mutation that they contribute to given trinucleotides that are listed down here. So deamination with age really focuses on these A, C to T, G mutations. That's what they cause for the most part. But then there's a few other ones here that are quite frequent. Apobec really focuses on TCA or TCC or TCG or TCT changing to Ts. Tobacco has a broader distribution of mutagenic effects. And this unknown clock-like signature, that's another aging signature, has a generally quite broad distribution as well. So we deconvolve that tumor into these different signatures to understand how much each one is contributing. That gives us a signature weight for every signature here. And I'm just emphasizing that, we can do lots of uncertainty analysis by bootstrapping the signature deconvolution. That's what all these dots are, many bootstraps on a given tumor, just saying how much of that signature do we really believe is contributing to that cancer. So we do that. And then you can also, and then in addition to understanding how much each signature is contributing to cancer, we look at the probability that each source created each variant. And we know that because we know what the sources are, and we can just look at the relative height of these bars essentially to give us the probability that each source contributed to a given variant. And then that probability comes out of that just by multiplying those together, essentially. And that gives us for P53 here, KLF5, and this odorant receptor, which doesn't have much cancer effect, what the probably each source contributed to creating each variant. And then we take that effect size that I just described to you, which is very high for this P53 variant, quite a bit lower for KLF5, but still there, and is basically non-existent for the odorant receptor mutation. So this is a really important variant, this is a less important variant, and this is not important at all. And then we can just sort of multiply through each variant by the probability that each source created that variant. And that gives us this final thing, which is the proportional mutation source effect size. That's a mouthful. But what we're just trying to say is how much of this given variant was caused by the particular mutational process. How much of the selection for oncogenesis was caused by that particular process. So the TP53 bars are much higher than the ones in KLF5, and those are way higher than anything in odorant receptor, because the odorant receptor, in fact, doesn't do anything for cancer. So the average attribute, then you can look across all of those, all of the variants, not just these ones, to look at what the average attributable effect size is in a given tumor, and you get this distribution which says, oh, for this tumor, most of the oncogenic cause came from deamination with age. And for this tumor, the second most common process that was creating mutations that led to oncogenesis was this light gray, which is this unknown clock-like signature 5. So this is a largely aging-driven tumor. And then there's a little bit of apobec here and a little bit of tobacco smoke, and you can follow it through like that. So this is one example for a given tumor. And then that result, it basically tells you what, at least with the knowledge we have right now, what the effect size by mutational source for this tumor was. This is a lung cancer tumor, by the way. Now you can look at this not just at, you can sort of understand that for a given site, but then you can also look at what a set of sites all look like. So this is just a diagram where we do that. Again, a little bit complex, but hopefully this everyone can follow along directly with. If you look across the genome, there's an average mutational weight. So tobacco smoke is causing a certain number of the mutations, a certain proportion of the mutations, and then a number of others are. And in these diagrams, I've sort of put the major mutagenic cause on the left and then stacked all the other causes on the right, just because it helps you really see the differential effect of these different processes. Tobacco smoking is the major cause of LUADs in general in terms of the underlying genomic mutation. But if you look at from site to site, each site has a different probability of being caused by tobacco smoke. So here's KRAS G12C, very, very, very strong cause by tobacco smoke. Maybe that's not surprising. In lung cancer, we see that variant very, very frequently. We very rarely see KRAS G12C in other cancers like pancreatic cancer, other KRAS-driven cancers. So why is that? Well, it's just because that site is hit a lot more in terms of mutations. It's not doesn't appear to have anything to do from our calculations with its particular cancer effect relative to other variants. And in contrast, here's EGFR L858R. It's a long known fact that you rarely see those in individuals who are non-smokers, are smokers. You see that in non-smokers. And the reason is it's not caused by smoking. So when you see a patient with an EGFR mutation, they typically are not a smoker. There's a lot more individuals coming in with EGFR who are not smokers than are smokers, relatively speaking. So you can do this for lung adenocarcinoma. You can look at other variants, of course. Lung squamous cell carcinoma. Here you see PI3 kinase, largely driven in lung squamous cell carcinoma by other effects, mostly apobec, but in fact, not at all driven by tobacco smoke. Again, that's an empirical observation that people have noted many times, that individuals with lung squamous cell carcinoma who have a PI3 kinase mutation are rarely are less frequently smokers than other mutations. P53 mutations, on the other hand, are diverse. Some of them likely to be created by tobacco smoke, some of them less likely. Okay, so we can look at this on an individual basis, and then we can look at some other cancers. So here's bladder cancer and cervical cancer. I just added these because maybe this is a little less well known, but a lot of the mutation in both bladder cancer and cervical cancer is caused, at least by this deconvolution approach, appears to be attributable to ApoBec mutation. ApoBec is this apolipoprotein B gene that enzymatically we know mutates DNA and appears to be a viral defense protein. And what we see is that a lot of the mutagenic cause in the genome is created by ApoBec. Some of it's by aging in bladder cancer and cervical cancer. There's a little bit of defective homologous recombination as a source there as well. But as you can see, for a number of these mutations, some p53 mutations for FGFR3, for KRAS G12D, we see almost no cause from ApoBec. But on the other hand, this other FGFR3 mutation, very likely to be caused by ApoBec. PI3 kinase, again, very likely to be caused by ApoBec mutation. Cervical cancer, the same thing. All right, so we can look at the individual variants here. Let's get back to the main theme that this talk hopefully is presenting to you, which is that once we understand for every one of these variants what the causes are and how much they're causing cancer, we can then look at tumor causation by tumor type. And this isn't the best way to contrast them. I'll show you another that maybe contrasts it a little bit better, but here we have all the different signatures on the y-axis and all the different cancers on the x-axis. And the red is the amount that the tumor type is caused by that particular signature, and the gray is, or black is, the amount that you see mutation for, due to that signature. And there's some big differences, say in signature 5 here for thyroid cancer, where you see an enormous amount of cause, but much less mutation. But it's a little hard to read that dot plot. Down below, we have just these bar plots showing the weight of mutation, how much mutation was caused, how much of the mutation in the genome was caused by a given mutational process, and on the right, the effects. And these may look pretty similar, but I'll show you the contrast that shows you how they're different in a moment. The thing I want to emphasize right now is we've given colors for all of those exogenous sources that may, in principle, be things that we could interfere on to stop. So UV light, defective homologous recombination, presumably, maybe there'd be a way to do that. ApoBec, perhaps if we, avoided viral infection. Tobacco, certainly interventional. Alcohol, definitely something we can intervention on. Mutagenic chemical exposures, definitely something we can do intervention on. Anyway, all those interventional ones are the colored ones, and the aging ones are the gray ones, and then the unknown processes. The processes that we haven't figured out what they're associated with are in black. So this diagram actually tells you a lot about what you can do now to understand more about cancer, right? Or to intervene. We can intervene a lot on these cancer types for which we see a lot of color. There's much less we can do for the ones we don't. So for instance, glioma, a lot of aging, not a lot of other things. Thyroid cancer, a lot of ApoBec, but other than that, aging. Glioblastoma, again, a lot of aging. Prostate cancer, a lot of aging, just a little bit of ApoBec and defective homologous recombination. So there's some we don't have much way to intervene on. Skin cancer, extremely easy to intervene to reduce the number of mutations. Lung cancer, a lot of tobacco, a lot of defective homologous recombination and ApoBec. So there's a lot we can do in terms of stopping those. And then also where there's a lot more to understand. So for instance, breast cancer, ER minus breast cancer, like nearly half of the mutations, we don't know why they're being caused process-wise. So this is something to be investigated. Because if we could figure it out, maybe there are these intervenable processes that we could do something about. Et cetera. So you can sort of look at the black, that gives you an idea of how much we still need to learn. And the gray tells you the idea of like how much more, how much aging versus other processes seem to be causing that given cancer. And of course, the cancers that are most age-related are at the bottom of this diagram, and the ones that are least age-related tend to be higher up. So this is just a bigger diagram of that same picture in case you can see it better. And then I'm going to show you, I'm not going to show you the actual cancer types, but just an animation that actually Vincent Cannataro made that helps you see the difference between the cancer mutation and effects. So this just varies between how much mutation is causing the given cancer and how much of the cancer effect by those mutations is causing the cancer, and allows you to sort of see how different they are. You know, for ones like skin cancer, it doesn't change that much because nearly all the mutations are caused by UV anyway. All right, so as I said earlier, the extent to which the process is contributed to tumorigenesis has been unknown, but now we can link it together with this process. And now I wanted to go back to this slide because I'm going to show you a bunch of diagrams, and they're pretty complicated diagrams, but on the left-hand side is going to be a bar plot that's reflecting like how much each process is contributing to the mutations. It's this left-hand sign. And on the right of each plot is going to be another bar plot that shows you how much each mutation is contributing to increased cellular proliferation and survival. For 4 different cancers. Here's primary skin cancer, primary and metastatic skin cancer, colorectal cancer, actually there's colon cancer, HPV negative head and neck cancer, and thyroid cancer. And the diagrams are this bar versus this bar. So the bar on the left is how much of for a specific tumor was contributed by a given process, and then how much of the oncogenesis for that tumor was caused by that particular process. And I've lined these up so that what I'm showing you is just 5 examples here from TCGA, and this is actually from some data gathered here at Yale on melanoma. But anyway, we've looked across these different cancer type, these different tumors, and the question is, are these different or are these similar? Like, is the basic mutagenic effect and the cancer effect similar, or is it different? And you see they're very similar for these two tumors, very similar for this third, getting a little different here, and getting quite different here. And these are arranged at the quartiles of the distribution. So it sort of represents the range of what you see in patients. So most of the time, mutagenic effect and cancer causation are aligned very closely in primary skin cancer, and that's because UV is causing almost all these mutations and changing things. In colon cancer, as you extend from the more similar to the more different, you see a lot more heterogeneity from patient to patient in terms of whether or not the causative factors are the same as the mutagenic factors. And that gets even more extreme with HPV negative head and neck cancer, and even more extreme with thyroid cancer. So, but let me just emphasize again, these measures are for individual patients. So in principle, this calculation can be done on any tumor sequence from an individual patient to tell you what the causation of their cancer was, at least to the level that we are able to analyze this. Now, there's a bunch of things that we would love to also be able to incorporate into this. This is only single nucleotide mutations. It doesn't take into account copy number variation. It doesn't take into account epigenetic changes. And as I said at the outset, none of this has to do with physiological things like whether you exercise and have good autophagy and it doesn't it's not that physiological question of why you got cancer, but it is the mutagenic answer of why you got cancer down at the SNV level. And so it reveals that, and so I think we're very good there. I would argue that the logic behind this is right, and that we can apply that same logic to epigenetics, to copy number changes, et cetera. There's just a lot of understanding we still need of the basic underlying mutation rate for those things in order to actually do that. And we're trying to work on that now. Now, I just, this is basically the end of the major talk, but I just want to emphasize that this doesn't just apply to the origin of cancer and the early tumor genesis. These same processes are going on in patients as we treat them as well. So there's, so if you have a patient where you take out a biopsy or a resection, and then they undergo some sort of treatment and have recurrence, there are ways to figure out exactly what the underlying processes that are contributing to the mutations that cause that recurrence are. So that should be of interest to all of us who are interested in figuring out what's causing recurrence in cancer. So there's a clinical as well as the sort of more public health side that I was talking about with regard to these mutations, the clinical side of how we might be able to apply this. And just to give you 2 material examples of this, here are 2, I'm going to show you 2 sort of tree studies of individual patients. These were led by Nick Fisk in my lab. Here's a patient who was diagnosed with stage 3B lung cancer. They had an EGFR exon 19 deletion, and their tumor was resected. They were given cisplatin and permatrexib. There's a little pie plot, and this is a phylogenetic tree relating their metastatic tumors to the primary tumor. And it's been dated. We have all these techniques in my lab to date that based on when the primary tumor was and how many mutations we see, et cetera, et cetera. These were extracted at a later date than this, and so that gives us a way to calibrate the time. And what you see here in these pie charts is I've made it simpler. I just am looking at all other kinds of mutagenic sources and one specific source that I'm interested in. And in this particular case, the source I'm interested in is the effect of cisplatin, which we know has a mutagenic effect on tumors, even as it applies its own selective effect, killing tumor cells. And what you can see here is that the cisplatin mutations on this branch, so this is independently determined, right? This isn't because cisplatin is here. We just did the deconvolution and boom, here are all these cisplatin mutations, this white pie piece here, almost, a bit less than a quarter of, or around a fifth of the mutations in this tumor are now cisplatin-derived mutations. And we can deconvolve that by doing this tree and seeing, okay, on this branch right here, that's how many are that kind of mutation. And then that, of course, that tumor continued to evolve. And the reason it continued to evolve, the patient was given erlotinib. Unfortunately, erlotinib wasn't very successful in this case because they got the EGFR T790M resistance mutation on this branch as well. And the tumor differentiated into these metastatic tumors and another metastatic tumor in the pancreas. And the point is here just that the proportion of cisplatin was discontinued. And so the proportion of mutations in subsequent branches is actually lower out of the total because new mutations are being added, but they aren't cisplatin related mutations. So all of this deconvolution and understanding of the underlying mutagenic causation occurs during this treatment process that patients receive, and we can figure it out. There's one more point that I just want to make here, which is that it turns out, and I don't have a plot for this, but the T790M mutation is a mutation that is very likely to be caused, just like those other ones I showed you, by cisplatin mutation. So this is a poor ordering clinically for these treatments to be given, because the cisplatin mutation creates a bunch of that genetic variation that is exactly what we don't want them to have if we're going to put them on erlotinib later. And very likely they had that mutation right when they were put on erlotinib, which is why there's a very little duration of benefit for the patient. So this is a great example for in terms of a clinical or exogenous source of mutation, the cisplatin treatment that they were receiving. Let me give you another example that's about an endogenous change that has an interesting effect in a very similar way. So here's another lung cancer case. This patient was put on erlotinib right at resection. They had a P53 mutation already after resection, but over a much longer period of time. They were never treated with cisplatin. A much longer time later, they did get an EGFR T790M mutation. And you can see these plots are solid here, meaning that the mutational process of interest that I wanted to talk about, hasn't happened at all here yet. And then you can see, unfortunately, later on, they were moved to Avastin, or not unfortunately, they were moved to Avastin. It wasn't unfortunate necessarily, but erlotinib was discontinued. They were given permatrexib and carboplatin late in therapy, but the divergence, but their metastatic tumors started genetically diverging about 2 years before their death. And the thing that I just want to emphasize is here's a catenin B1 mutation, S37C, which is known to induce defects in homologous recombination-based mutations. And you can see in the descendant lineages the increased amount of that kind of mutation occurring after catenin B1 mutation. So this is an endogenous process that was started by a mutation that we can then track again down to the individual branch where the mutations are occurring and how many of them are caused. And then from then on, there's a lot of catenin B1 mutation in these tumors, but not in the spleen. That's an interesting story here is that this catenin B1 mutation occurred and led to all the metastases to all these other tissues. The one tissue that had a metastasis that was not cadherin B1 mutated was the spleen, which is very interesting because cadherin B1 mutation causes vascularization. The spleen is already quite highly vascularized, so it may not have been needed for the spleen, whereas it may have been more important to the cancer for the rest of the tumors. All right, I've sort of gone through all of what I wanted to talk about today in terms of introducing you to this way of actually trying to understand, why an individual tumor has been made oncogenic. I hope that I've at least been able to argue that the logic behind what we're doing is sound, and that the process that we're doing is a sound way of sort of attributing that cancer effect, at least as regards those single nucleotide variants, which are what we mostly focus on, but where we don't know whether that's 10% or 90% of the reason why genetically tumors are oncogenic. We don't know that yet, but regardless, if we look at that single nucleotide effect, we now can sort of deconvolve that. And I'm very curious if anyone has thoughts to share with me, how this information could be used for the benefit of patients, for the knowledge of patients, but also, as I mentioned in the later part of my talk, in understanding better what our therapies are doing to patients over time as well, and ways that we can ideally order our therapies so that we avoid the evolution of the resistance that we're trying to avoid, which is so clearly evidenced in that one case with the cisplatin and erlotinib treatment. Thanks very much for your attention. I want to thank very much Vincent Cannataro, who was a postdoc in my lab, is now a professor at Emmanuel College, remains a vibrant and wonderful collaborator of mine. I really like working with him. It's been incredibly productive to continue to do so, and I'm really delighted that he's been able to do so despite a very heavy teaching load at Emmanuel College. Jeff Mandel over here in the audience, graduate student working on the cancer effect size calculations and the machinery underlying that. Nick Fisk, who's worked on a lot of the tree-based analyses in my lab, including those last ones that I mentioned. Everyone else in the Townsend lab, they're also great. I didn't specifically mention their work. We've got interesting work on epistasis and all kinds of other things that that I really think is outstanding and should be really interesting. So all the members, it's a great group. Also, I want to thank the NIH, NIDCR, Yale Sport and Head and Neck. It's a great community, and I really enjoy being part of it. And it also provides a substantial amount of my funding for cancer work. So thank you very much, and I would love to take any questions or hear any thoughts, comments, etc., from anyone in the audience.
Jeff, I think you actually have an amazing talent to make really complex things, explain them in a very simple and logical way. Do we have a microphone so that people could actually ask questions? Oh, okay.
Amazing work, Jeff. I just wanted to ask if you could address how you handle co-mutations when you look at cancer effect size. And, I'm thinking of this finding that we have in head and neck cancer where P53 mutation is truncating if you don't have CDKN2A mutated. And when you have a mutation in the DNA binding domain, it seems like you need the second mutation. So how do you handle that? And then I guess also when you have P53 mutations or CDKN2A mutations or whatever, does that muddy your signatures at all? Do the mutational effects of losing control of cell cycle and DNA repair
So
show
let me
up?
answer the second part first, because it's a little more straightforward. Does it muddy things when things get changed? Yes, it does, in the sense that there's a temporal difference, right, between what was happening before that change happened and what happened afterwards. And our resolution for understanding those temporal differences is somewhat weak, right? So generally, if we have a lot of samples like we had in those two cases, we can sort of piece apart when things happened in a nice way, and we'll be able to understand those sorts of differences. But when we're just looking at tumor genesis to resection and we have this association, then we have to use very large numbers to get sort of statistical associations to understand that sort of ordering process. Which gets me to your second question, or your first question of co-mutation, which I have a strong opinion on everything. So my strong opinion on this is that the general genomics approach towards looking at co-mutation is flawed in a way that is not apparent when you read all the papers on And the argument that I want to make is that when you look at co-mutation, you're typically looking at a very observational thing, which is like, how often is this one mutated and this one mutated? So for the same reasons that I outlined in my talk today, that there are two reasons why you see things mutated, underlying mutation or selection. There are two reasons why things might be co-mutated. They might be co-mutated because when you get one, the other one is selected, and it really creates a great benefit to the cell to survive and replicate. The other reason might be because they both have the same underlying mutational process. And when you have 4 orders of magnitude of difference in mutational process site to site, that can be a very big reason why you see co-mutation. So co-mutation is not the signature we'd like it to be to say these things are selected together, because sometimes they may not be, even though they're strongly co-mutated in a data set. So then how do I deal with it? Well, we can take all of the approaches I told you, and we're working on even more sophisticated approaches now to try to do this. I think I have some slides on it, so I would love to take the time to just quickly introduce it, since they're way down here though. Yeah, so this is the point that I was just answering to you, which is mutual exclusivity and co-occurrence are patterns that are caused by either co-mutation or what I call selective epistasis. Again, I'm using the terminology from my background in evolutionary biology. Epistasis meaning one gene is having an effect on another, or the mutation in one gene is having an effect on another. So typical approaches have not acknowledged the possibility of co-mutation, which is a common underlying mutational bias. That's what I just said to you. This is a typical slide from, I don't mean to be, casting aspersions on this. As I said, this is what everyone pretty much does, but they look for whether cancers have sequential mutations developed or co-mutation. But we can actually take those same analysis, the same data, and deconvolve with some fairly sophisticated mathematics that Jorge Alfaro Murillo and I did on the fluxes, mutation rates, and scaled selection coefficients for up to 5 genes, and look at what the likelihood of individual genes are to get mutated, what the likelihood KRAS is going to be mutated after P53, et cetera, et cetera. We can look at all of these, figure out how frequently those happen. So this is the flux, which is a measure of co-mutation essentially, the underlying mutation rates, and then the scaled selection coefficient for the new mutation. So these are how likely is P53, how likely is KRAS to be mutated after P53. And then how likely is it, how selected is it to have KRAS after P53 as a separate measure. So we can basically take all of those and look at all of those different things for up to 5 or 6. And again, our constraint is usually the amount of data. We need massive amounts of data to understand like 3-way effects or 4-way effects. So you need to have examples of every possible combination in that data set, and that rapidly exhausts our samples. But on the other hand, we're getting a lot more data now. And so we're able to do this with more and more data sets now. This is lung cancer, and we were able to do it for these 5 genes, P53, KRAS, STK11, LRP1B, and figure out all their relations with each other. This is maybe an easier way to see this instead of a big table, which is just what's the evolutionary trajectory of them. And again, this is all epistatic, like it's all taking into account that co-mutation factor. And the width of the bar is the flux, or how frequently you go from normal to say P53 in this particular case, or LRPV1 or KRAS or STK11. And then you can see that if KRAS isn't actually that frequent as a first mutation, but if you do get it, then you're very likely to get LRP1B or STK11. If you get P53, you're very likely to then get LRP1B as well. You have some probability, but it's not so high of getting KRAS after that. You're very likely to get a KRAS mutation if you have P53 and LRP1B together, et cetera. So you can look at what the likely trajectory for a given patient is. You could even look at where they are on this trajectory, and we haven't done this, but presumably you can figure out what their prognosis was based on where they were on this diagram, et cetera. And we have basically a map of what's actually happening to these patients. And then down below in the smaller diagrams, I've just divided this up because this is all the fluxes again, let's divide it up into mutation rates and selection coefficients. And what you see is the mutation rates are quite symmetrical because we haven't accounted for things like Cadherin 1B mutation changing the mutation rate, et cetera. In this particular analysis, although in principle we can do that. And then on the right are, so there's a LRP1B particularly has a very high mutation rate, so its relatively high frequency is not that big a deal, although it does seem to have some selective effect as well. And then over here we see the selective effects, and you can see there's very strong selection for P53 initially is the major selection, and that exists after LRP1B as well. But after after P53 or LRP1B and P53, then we're very likely to get this KRAS mutation, et cetera. So you can really understand what the relative effect of each of these is.
How stable these trajectories are with the sample size that you analyze?
Yeah, that's a good question. I haven't done the study that I'd like to do to answer that, which would be to like do some very massive analysis. It's actually a lot of computation to like do that 1,000 times, subsampling, et cetera. But what I have done is just do the analysis, with one data set and then add more data sets. And it seems quite stable from that perspective. That's not really the same because we're not subtracting out the first data set when we do that, but it's not like it varies all over the place. And the stability, of course, is proportional to the prevalence, right, of that particular mutation. The mutations that are really highly prevalent, they stay very stable because we've got a lot of examples of them with the other genes. As soon as you get to lower prevalence, it's a lot iffier. So really, this can only be used right now for the most prevalent kinds of mutations that you see. And typically, we are, for instance, assembling all the mutations in a given gene as one kind of mutation, because we need that sample size to do that, which is something that in my other research I usually avoid because I think it's really important to understand that different sites have different effects.
So one thing that I didn't see, which sort of you probably did this, so you can calculate the mutational process contribution to the driver mutation in particular individual cases. But what happens if you cluster cases? And obviously you should be able to tell apart lung cancers related to smoking and those who don't. And that would be a trivial thing to do. But could you do the same and create a new classification, for example, for endometrial cancer or breast cancer that are related to aging and the other group? And by looking at them separately, you might get some idea about what's actually causing the unknown mutation signature. Like this is the cluster in the cases.
Yeah, I definitely think you could cluster them. I think, you're reducing the dimensionality of the data when you go from the raw data back to the processes. And so you have a reduced dimensionality of that raw data. And then if you were to cluster on the basis of this, you would be taking that reduced dimensionality data and trying to say, does that predict something? So I think from a machine learning standpoint, you might want to just go back to that raw data in some way. But there might be some way of thinking about it. That I say that, but then I also think there's a second part of that, which is that I do think you do better looking at actual biological processes, even if it involves some reduction of the data, because it simplifies the data in a way that means you don't go off on these random tangents of all the noisy stuff you're looking at. So I guess there's a tension I think you should be wary of in doing that, but I don't see any reason you couldn't do that. And it would probably be highly predictive in some cases. You're probably going to see most skin cancers very easily, predictive that way, because they're just UV all over the place. Some other cancers are probably quite hard to distinguish one from the other just by the mutational processes that underlie their cause. And so I could imagine doing that. We haven't done anything like that. Any other comments?
Oh, yeah. Is there a way to ask questions from the audience online?
There was, I thought, Q&A, but there it is.
Do we have time?
Oh yeah, we've got some questions here, but maybe one more for you and then I'll go to the online questions.
Yes, okay, thank you. Thanks, Jeff. Fantastic work. I think your methodology is on the right track. Nothing to worry about at all. The opposite is true. My only concern is availability of data in the future, especially for new types of cancers. Are we asking the right questions? Are we collecting the right data? We meaning us humans. And I'd like us humans to ensure that this data is available. It's open source and it's reliable. And what are your thoughts on that?
Yeah, so that's a great question. I mean, I think that the volume of data sets on like tumor genesis resection kind of data is going to increase very well on its own. Like we don't need to pay attention to that question. The data sets that I think I would like to see more of are these multi-sample data sets from individual patients. Back in 2016, I was lucky to be funded by Gilead to actually sequence these large numbers of metastatic and primary tumors, and they were really, the potential of those data sets is really high. especially if they have the clinical annotations alongside, so you can map it to understand what was happening for the patient at the same time as what was happening genetically. That data set, though, was heterogeneous by cancer type, right? And I haven't seen similar sized data sets on individual cancer types gathered. And it's not, it's a lot of money, like a couple $1 million to do that sequencing, but you could do that for every cancer type for, $30 million or something like that. And I think that would be so worth it because we would learn so much about the evolutionary trajectory of each of these cancer types by looking at multi-sample data like that. But I haven't managed to sort of put together the argument to get funding to do that.
I encourage you to elevate that, definitely.
Yeah, thanks.
Could I comment on just the comments you made?
Actually, okay,
First, I enjoy your talk,
thank you.
but I'm not so sure. Even the tumor heterogeneity, you map just the tumor cell. We don't even talk about the microenvironment. Mass sequence will really be useful without the other tools. In other words, you're going to, for instance, you just mentioned about the cluster approach. You can have a mutation in different tumor cells within the tumor mass. When you do the analysis, you put them all together. Does that make sense?
I think I might need to talk to you at more length to sort of fully understand your question, but I guess what I would comment is just that when I say this is the kind of data we need, I'm mostly talking about for the kind of work that I'm talking about, rather than for everything to solve cancer, of course. So, but in order to understand the underlying selective coefficients and understand the mutational processes, I do think large amounts of tumor to resection data, which will be gathered anyway, but also more of this multi-sample data so that we can understand dynamically over time what's happening, which we can do, I said, in a probabilistic way, but never in a very satisfying way with just the tumor genesis to resection data.
Yeah, I think this relates to the noise that the tumor cellularity differences we bring in. And I think it also relates to Kruger's question about the copy number changes. So how do you adjust for this? That, if a gene has 17 copies, whatever mutation it has, that signature is going to be amplified. And it's not necessarily reflect the actual 10 times higher prevalence of contribution of a particular mutation process, but it's just that the gene amplified. And the same, I think, these questions about the tumor cellularity. So the adjacent normal tissue also requires mutations, and they actually introduce noise, right, of some sort.
Yes. So both of those are sources of noise in the sense that on average, as we look at, so talk about a gene amplification, for instance, is a great example. When you get a gene amplification, the mutation itself may not be contributing the cancer effect size that we analyze when we get this kind of data. But what is true is that those mutations and the amount of copy number amplification that they typically have contributes this amount, because we're just looking at whether or not we see these in patients, and whatever other processes are going on, we're averaging over. So the cancer effect size is still, I would say, still the measure of how much that mutation is contributing to it, but the means by which it contributes, we don't really know from this analysis. It's just that wider question of how much is this variant contributing. And if it needs amplification as part of that process, well, then we need to do a more detailed analysis that looks both at amplification and the mutation. And then we'll be able to say like how important that mutation is in terms of cancer effect, how important the amplification is in terms of cancer effect compared to the mutation itself. That's not something we've been able to do yet, but it's something on our agenda. It's very difficult, but I think it's achievable, but very difficult. I think I better quickly ask, I feel sorry for the people who ask questions online. The one question is mutation a biochemical reaction to T or G or C, a substitute of T or G or C? The mutations I'm talking about in this entire study were all single nucleotide mutations in the context of what I meant by trinucleotide context is there's 3 mutations and the central one, how is that mutated to another single nucleotide? There are ways to look at doublets. There are ways to look at some other more complicated indels, which we have in the lab almost implemented, but other mutation types we don't have actually looked at. Yuval Kluger's question, I think, thank you. You have echoed that for me, Laos. So I believe I answered that. that, basically it's true that we don't know the specific, when we talk about this mutation and how much its cancer effect size is, that's in the context of everything that happens to that mutation in cancers, and it's the average across that. Tim Robinson has a question, which is, can the spectrum of mutations tell us about the chance that a tumor will respond to treatment? It may. Well, so for instance, the fact that there were cisplatin mutations is going to tell you that it's likely to have an EGFR T790M resistant mutation sort of sitting there waiting to come out when you give erlotinib. So in a sense, that spectrum could tell us about the chance that a tumor will respond to treatment. But in general, if I could, I would rather look for EGFR T790M itself directly. For example, if the vast majority of mutations are in melanoma are BRAF V600E, and the vast majority of cancer causation by mutation is there, does that inform the chance that the tumor will respond to directed therapy to BRAF? I think the number of mutations I don't think does at all. I think that what's important to understand about BRAF V700E and its cancer effect size, which by the way is a very high cancer effect size, is that if you can get a therapy that treats BRAF V600E effectively, it will be a very effective therapy. And there's a good example of that, and there's a caveat to that example also, which is that BRAF V600E, as many people know, there's vemurafenib, which is a very effective therapy for skin cancer. The only problem is there's very rapid evolution of resistance. Nothing about cancer effect tells you how quickly resistance will be evolved. And in that case, this also interplays with CNVs, because at least one of the explanations for why that rapid resistance occurs is that you get amplification of the variant BRAF V600E that basically overwhelms the treatment of vemurafenib and means that and that's a very fast process. Amplification of a gene in a genome is not hard to do, as a high mutation rate happens very quickly. Some cells have more of it, some has less, and those ones with more are selected. It's very easy to select on that basis. So, I think it informs you about how likely a treatment is to have a big effect at the moment you apply the treatment. How quickly you evolve resistance is another question. And R.D. Mustaki, the sources of mutations, smoking, UV, infection, affect the normal non-transformed tissues. Yes, they do. Can you use your approach to calculate the cancer effect that mutations on the tumor microenvironment have on tumorigenesis? One might argue that a lot of these mutation sources act on the environment, reducing the fitness of a normal cell, allowing the cell to grow. This is a really interesting question. We are working on this. So the bottom line is that, and I'll be very quick with this answer, that once we are able to figure out these cancer effects, then we can ask, to the extent that we have annotated data on this tumor was exposed to this given environment, we can ask, how does that environment affect the cancer effect? So we can ask, if you have different ages, not just what mutations are caused by aging, but how much does the cancer effect of a given mutation change as someone ages? So there's ways to do that with the kind of data we have. Again, it requires bigger sample sizes in general, but we're looking at that right now with regard to smoking, because smoking, of course, can have a direct effect of mutating individual genes, but it can also have a physiological effect of degrading the normal cells in general in the lung ecosystem. And because you have degraded normal cells, that could increase your chance of getting cancer, or it could mean that certain mutations are more likely to be able to make cancer proliferate and survive better than other mutations. So the physiology could be very important, and there are ways to get at that. But you need to know this first, and then you can ask the question about physiology affecting things. And I think I'm out of time.